Hello, everyone, and welcome to today's webinar, Catapult Your Cell Therapy Research into the Clinic, CAR, NK Cells, and Tregs. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit www.miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speakers will answer questions via email to the address you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Julia Hengst, expert for NK cells, Miltenyi Biotec, Dr. Martha Elia Luevano Salinas, expert for engineered cells, Miltenyi Biotec, and Dr. Alina Steinbach, expert for cell sorting, Miltenyi Biotec. Speakers, you may now begin your presentations.
So now welcome to the part from the Translational Cell and Gene Therapy mini webinar series about the CAR-NK cells from research to clinical NK cell engineering. So in the next 20-25 minutes, you will learn more about NK cells and how to engineer CAR-NK cells and how to translate this into the clinical setting. Here's our disclaimer. And we will start with a short introduction, then I will present the research workflow. My colleague Marta will focus on the clinical workflow, and then we will summarize our resources and you can ask questions. So first of all, why to use CAR-NK cells? So in addition to, for example, CAR T cells. So NK cells and more recently CAR-NK cell immunotherapies has been shown in several clinical settings. And the advantages is firstly the superior safety because the risk of on-target or off-tumor toxicity to the normal tissues is relatively low due to a limited lifespan of CAR-NK cells in the circulation. And allogenic CAR-NK cell infusions have reduced risk for graft versus host disease. And also, in addition to that, the cytokine release syndrome and neurotoxicity are less likely to occur in CAR-NK cell immunotherapies, partly due to a different spectrum of secreted cytokines. So meaning that activated NK cells usually produce interferon gamma and GM-CSF, whereas CAR T cells produce different cytokines, including, for example, IL-1, IL-2, interleukin-6, TNF. So because of this different spectrum, the CAR T cells have a higher or are more highly associated with the cytokine release syndrome. Then, in addition to that, there is a killing mechanism. So the CAR-NK cells can, of course, induce the CAR dependent killing, but on top of that also can kill target cells in a CAR independent manner, as they have the natural capacity to kill tumor cells. So they still have this capacity and can, for example, be activated by different receptors, so by natural cytotoxicity receptors or NKG2D or co-stimulatory receptors, including DNAM, or by, for example, activating KIRs. And in addition to that, NK cells can eliminate tumor cells through CD16-mediated antibody-dependent cellular cytotoxicity, so ADCC. So they have a lot of potential to kill their target cells. And because of that, they are a very good source or allogenic source. They can be generated from multiple sources. And there is no need to produce a CAR product for each from each patient for themselves, so in an autologous manner. And then just to highlight how the CAR-NK cell trials have been increasing over the last year. So there has been a very, very strong increase, and different starting materials have been used. So there are iPSC-derived CAR-NK cells, or cell lines have been used. Cord blood can be used to engineer NK cells, or also peripheral blood. So there's quite some variety on the starting material. And now looking at what Miltenyi Biotec offers for the CAR-NK cell engineering workflow is that we offer everything from research to clinical manufacturing throughout the whole process, starting with the cell product in the beginning. So where usually it's either cord blood, peripheral blood, or, for example, the PSCs, as we heard in the talk before. Then the first step is always to isolate the NK cells, to activate them, to transduce them, and then afterwards expand them to reach the cell numbers which are needed for treating the patients. And of course, the verification, but not only at the end of the process to really assess the functionality of the CAR-NK cells, but also throughout the whole process to ensure that the whole process is working and that the engineering is taking place in the way it should be. And thus we will focus today both on the research and on the clinical side and show which products we have here for the workflow and how this is working. So now looking at the translational workflow, meaning offering everything from research to clinical. In the first part, we will fully concentrate on the research side and then show that all the reagents are also there as GMP and how data looks like from this. So now looking at the research workflow and on the first step, so the cell separation, where the NK cells need to be isolated. The isolation process is first in the first step, after, in this case, having your PBMCs, the CD3 cells are depleted with CD3 microbeads. Then the negative fraction is used to enrich the CD56 expressing cells, so to really have only the pure NK cell population. And after that, it's very important to activate the NK cells and to expand the NK cells. And for both these steps, we have the NK max medium, which is used in combination with the premium grade cytokines IL-2 and IL-15. And here I'm just showing one graph where you can see from isolated NK cells, the NK cell expansion with the NK max medium in comparison to competition. So there's really superior expansion of the NK cells. And also it has been shown that the NK cells, so now I'm just really talking about NK cells, keep their cytotoxic potential after this 14 days of expansion. And here you can also download the full protocol. I'm highlighting this here because it is very important to have a good activation and expansion protocol for the NK cells prior to the transduction. So the NK cells get activated first and then expanded in the medium. And here you can now see the comparison of NK cells being cultivated in the medium and CAR-NK cells. So showing that also the CAR-NK cells can nicely expand with this combination of medium and cytokines. Just to mention here, what is very important also when thinking translational later on, that these premium grade cytokines have a lot specific activity. So this can be calculated and thus being applied in a reproducible manner. And all of these reagents are available also in GMP. And just to highlight the medium once more, it has been used already for NK cell expansion, but also with regards to CAR-NK cell engineering. So there's a lot of, or there are some references and some proofs that this medium is really widely used by the community. And now looking at the transduction, which is also very important because, of course, the higher the transduction, the more starting material after the transduction step is there to expand. What you can see here is on the lower line, the primary NK cells, and here a cell line of NK cells. The first row shows cells which have not been transduced, then NK cells which have only been transduced with a vector, with a baboon envelope lentiviral vector. And then here, the last row, where vector together with vector fusin has been used. And you can nicely see that the transduction efficiency increases from 17 or 20% up to 80 to 90%. Just some more background information for vector fusin 1. It's a viral transduction enhancer and can enhance lentiviral and retroviral transduction. It is fully synthetic, non-toxic, and soluble peptide. And also one important note, it is very easy to apply as no pre-treatment or pre-coating is needed. So you can just directly add it to the viral supernatant. And also this is available in GMP quality. And here you can just see another plot where NK cells have been efficiently transduced. And here you can see the CAR expression of the NK cells. And now to the final, my final point, which is the cell analysis, is that, of course, at the end and throughout the process, you want to verify your NK cells and CAR NK cells. So the max quant in combination with our reagents allows to define the cell count, to look at the cell fitness, the composition of the cells, and also to analyze the intracellular cytokines. And in addition to phenotyping, of course, it's also very important to look at the function of the cells. So for this, there is the need for co-culture experiments with specific target cells. And here then the efficient target cell or the target cell killing can be analyzed, and also the secretion of cytokines. And just some more information regarding this. So to detect the target cell killing, the target cell line will be co-cultured with the CAR NK cells. And then in an effector to target ratio dependent manner, of course, the killing capacity is analyzed, and all of this can be done in combination with the max quant analyzer. And then, for example, to analyze the target cell killing, PI can be used for live dead staining. And then here you can nicely see the killing of the tumor cell line, which expresses the antigen of interest, which is recognized by the CAR NK cells, and then the control cell line. And that this is, of course, then effector to target cell ratio dependent. And in addition to this, there's also the analysis of the cytokines possible, where up to 12 cytokines can be analyzed within one sample of supernatant, and only 50 microliters of supernatant are required for this. And in combination with the max quant, this can be analyzed. However, the maxplex kits, which includes these 12 cytokines, can be used with any flow cytometer. And here, out of these 12 cytokines, 3 are highlighted, where it could be nicely shown that after co-culture with the target cells, the CAR NK cells have an increased, or secrete an increased amount of GM-CSF, interferon gamma, and TNF. And with this, I'm coming already almost to my last slide. So to summarize this, because the in-process control and the quality control is very important. So for this, we offer the reaffinity antibodies for very specific staining, and also there will be predefined antibody panels for the CAR NK cell analysis. There's the CAR detection reagent, and what I just showed on the slide before, the maxplex cytokine kits for very fast and easy functional cytokine analysis of 12 cytokines at once, which can be easily analyzed with the max quant analyzer. And then there are the express mode package for packages for automated and user independent analysis. And just to highlight this, that our solutions are developed throughout all stages, so fully translational. What I showed you for the research part, now there's the possibility to do the mouse, which I didn't cover with this presentation, the human and also the CAR screening possible with our flow cytometry solutions, and then with in the preclinical setting, humanized mice and process optimization is possible, and with clinical trials, the quality control monitoring and concomitant research. And mentioning the clinical trials, I want to hand over to Marta for the clinical part.
Thank you so much, Julia. So I have just a couple of minutes, so I'm not going to be too long. I will just briefly show you what would be the translational application of what Julia just did. So you just learned about how to manufacture CAR-NK cells on the research setting. And for the clinical part, the good news is that we offer all of the reagents that you're using in the research workflow in a GMP grade. And this is very important because Julia and I just came back for a customer tour in the US, and we learned that a lot of the researchers are really not thinking ahead. So most of you that are doing NK cell research are really thinking about translating that into the clinics. So it's very important that you already know, or at least think about what type of workflow will be needed in the clinic, and to make sure that this is compatible with what you're doing now. For example, the use of feeder cells. This could be difficult in the future. Or right now, you're using the NK cell isolation kit instead of the reagents that are needed to really separate in clinical grade, like CD3 and CD56 microbeads. So just keep that in mind. I don't want to spend so much time, but whenever you translate your research into the clinic, it is a complicated process in which the cells have to go through different steps. And all these different steps needs to be done normally in a manual setting, requiring quite a lot of equipment. And also these open steps are risk because you can contaminate your cells. But we have designed a process, the Clinimax Prodigy NK cell transduction process, which allows you to close the system and do everything automated. Chao explained to you how the Prodigy works and what are the specific characteristics of this instrument, and we have taken advantage of this to present to you the natural killer cell transduction system. So what does it do? You will have your leukapheresis product. It will go through a step in which you will deplete the CD3 cells. Then later you will enrich and transduce in one tubing set and do the expansion there as well. So this is pretty much the workflow, including all of the different reagents in GMP grade and also protocol that we have standardized and we have already proved. So the next couple of slides, I'm just going to show you some of the results. We have different webinars as well for this and more in depth, but just in general, I want to start saying that we are using a different lentiviral vector envelope because we tested the VSVG, and this is the most common one used for the T cells, but we learned that this is not working for the NK cells. Therefore, we changed that and we're using the baboon envelope. And together with the different activation, like interleukin 215 or the interleukin 1 family cytokines, we're able to have very good results. So just to give you a brief overview, once you complete the selection procedure, which is the depletion of T cells and enrichment of CD56 cells, using this procedure, you can get very high purity and very high viability. We do a transduction at day 2. Every protocol is different. You might be doing something different, but it's fine. We have learned that using vector fusin 1, like Julia showed, enhances this transduction, and we're using a spinacylation, and also in our hands, this enhances the transduction rate. We use an activation with interleukin 1 family cytokine just in the beginning, and we remove it at day 3. And you can see in this slide that we have a very nice stable transduction rate at day 7 and then at day 14 when we end our culture. We have observed that the NK cells in the first 7 days do not grow. Therefore, after day 7, you can see an exponential growth. And this exponential growth is not only for the NK cells, but also for the CAR NK cells. And we show that the percentage of CAR NK cells remains very stable during the culture, and we are doing 14 days. At the end of the 14 days, we have a very pure NK cell product, very good viability, and most importantly, very little contaminant cells. And this is highly important because we're using NK cells in an allogenic setting, meaning no threshold or there's no opportunity to have any contaminant cells. We have a very high amount of NK cells. These are around 1 × 10⁹ of total NK cells, and being around half of them CAR NK cells. We're also developing another process with a bigger chamber to have higher amount of CAR NK cells. We have tested those cells and they're functional. So if we put or co-incubate NK cells with either a cell line that doesn't have the target or has the target, the non-transduced cells, there is a baseline of killing, but the CAR NK cells are able to kill efficiently the ones that have the target. As Julia mentioned before, we also measure the activity of the cytokines release, and we also are proving that the NK cells are able to secrete high levels of interferon gamma and GMSF, and these are very specific for the target, and very important to compare here to the CAR T cells, because the T cells are known to cause this cytokine syndrome release. We have, at least in this experiment, shown that the CAR NK cells release less interferon gamma, which is also relevant. So this is just a snapshot and a summary of what we can offer. Most importantly here, I think you can save the hands-on time. Maybe you are not in the clinic yet, but just think about whatever you're using for your research, it will be translated into the clinic, and you want to have reagents and procedures that are automated so they can be robust and reproducible. If you think that you don't have enough CAR NK cells, we're also offering via their customized application team the opportunity to connect these to bigger systems like the G-Regs or also the Wave, for example. Also, other approaches are really generating automatically the NK cells or CAR NK cells, do a cell banking, and then later on use a bioreactor. To finalize, the same as Julia just mentioned, we need to have very strict IPC/QC during the manufacturing, and we have developed already the panels for this and the detection reagents for this. We have a protocol that it's available for you in which the most important parameters are there. The panel is very simple, but it's necessary to have your release criteria. So the contaminant cells should be low and your purity and your viability should also be high. We have CAR detection reagents. Mainly, these have been very famously used for T cells, but they also work for NK cells. So there are some type of CD19, for example, therapies around that the T cells are using, but also NK cells are doing that. And we have also developed different CARs for other like CD33 or BCMA. And we have tested this not only for the T cells they also work for the NK cells. So if we're using any of these types of targets like CD19, you can also use our CAR detection reagents. And just to finalize, we have plenty of resources for you. We have the complete protocol for CAR NK cells. We also publish that, so you can check out the publication that we have. We have in our website the application protocol with every single step, the timeline, all the products, tips and tricks, and all the data. And of course, if you have any questions, make sure that you let us know, and we're happy to answer right now in the few minutes that we have any question. Thank you so much for your attention.
Hi, my name is Alina and I'm a product manager for the Max Quantito, and I'm now really happy to talk with you about how you can advance your clinical Treg research. This is the agenda for the talk. So first we will have a look into some introductory words to regulatory T cells. Then we will talk about where Tregs are actually used as a tool. And then we will have a look at different workflows that can be used to manufacture clinical Tregs. And in the end, of course, we will also have some closing remarks. So Miltenyi Biotec is at the forefront of pioneering clinical research, and this is focusing on immunotherapy, graft engineering, and also tissue regeneration. We provide different platforms for that, such as the CliniMax Prodigy platform, but also the Max Quantito cell sorter, and we will have a closer look at these two systems during the talk. So first of all, let's sum up what regulatory T cells are actually doing. They have a really pivotal role in the immune system. They have three main functions. The first is to suppress overwhelming immune responses, and by this, Tregs are keeping the balance and the homeostasis in the body. The second function is to also suppress immune responses against self-antigens. And by this mode of action, Tregs actually protect against autoimmunity, and they are mediating the tolerance. The third mode of action is unwanted, but I will name it here because it's very important for cancer therapies. Because actually Tregs, because of their mode of action, they can also suppress anti-cancer immune responses and thereby support the tumor growth, which is of course what we don't want. Let's have a closer look at regulatory T cells here at the bottom. They don't have a unique marker. They are identified by the expression of CD4 and CD25, but also by the absence of CD127. And there is also an internal marker, which is FOXP3. In the end, Tregs are classified into three main subtypes, and this is based on where they actually come from or where the cell type has been developing. And this is naive Tregs, peripheral Tregs, and there is also in vitro or induced Tregs. So Tregs are an attractive tool in the treatment of different diseases, such as inflammation, autoimmunity. They are important in transplantation or graft versus host disease. And as I said, also they have to be considered in cancer. In the healthy body, there is a good balance between regulatory T cells and the effector T cells. However, there are situations during a disease when this gets into some imbalance. And for example, here it could help if the Treg compartment is replenished with a Treg infusion to again restore this balance. So therefore, Tregs are an attractive tool, and they are already used for graft versus host disease, autoimmune diseases, and also in transplantation. The manufacturing of clinical Tregs is a challenging task, and there are different steps that have to be considered. The first step is the isolation, which is of course very important and has to be contamination free. Another step is that Tregs are usually needed in an ultra pure state to have an effective treatment. And also, of course, the maintenance of the cells in culture is very fundamental to the whole process. The good news here is that Miltenyi Biotec can provide solutions for all of these pain points. We do have the platforms to effectively isolate the cells on the CliniMax Plus, CliniMax Prodigy, and also the Max Quantito cell sorter is helping in these regards. And for the maintenance of the cells and the culture, we offer different reagents as well. So let's then jump to the exciting part and have a look at different workflows that we can offer here to manufacture clinical Tregs. So Treg manufacturing is basically composed of 3 steps. First, the isolation of the Tregs, then the Tregs usually go into an expansion phase to get meaningful numbers for a treatment. And then after that, of course, one has to analyze the cellular product. For all of these different steps, Miltenyi Biotec is providing suitable products. So this is showing the steps in a bit more detail that I will also explain. And we do have for the Treg isolation 2 workflows, workflow 1 and workflow 2. And in a minute, we will have a closer look at them. So for workflow 1, the long-known Clinimax Plus platform is used. It is taking over the depletion of CD8 and CD19 and the CD25 enrichment step. And then it's optional also to use the max quantitative cell sorter if a Treg subset is being looked for. This workflow is used in graft versus host disease, and here the aim is to get intermediate to high purity Tregs out of it with around 96% of purity. The whole isolation process takes 7 hours, and the expansion phase, depending on the requirements, can last from 12 up to 36 days. However, in the recent years, there were some pain points that were popping up, and more requirements to Treg manufacturing came also up. So researchers are more looking into Treg subsets, such as naive Tregs, and this is a really small cell population, but on the other hand, one needs high numbers for an effective treatment. And on top of that, the need for ultra-pure Tregs is also rising. GMP flow sorting is also a challenging task, and it's hard to get approval for clinical studies. So teaming up the Clinimax Prodigy and the Max Quantitative Cell Sorter is the perfect fit to address these pain points, and we will have a look at this on the next slides. So this is workflow 2 that we can provide, and this is based on the Clinimax Prodigy LP25 process. And first, what is done is a CD25 enrichment step. After that, the Tregs get labeled and they get eluted into sorting buffer, and then they are further sorted on the Max Quantitative Cell Sorter. This workflow is used in autoimmune diseases, in CAR Treg, and organ transplantation, but also graft versus host disease. And the aim here is to really get out ultra pure Tregs with a purity of more than 98%. The isolation process in this case takes 10 hours, and there is again a expansion phase between 12 and 36 days possible. So let's have a closer look at that. So here we can see that the naive Tregs are actually a really tiny population of the white blood cells. And when we look at the gating strategy, we see that with starting of the cellular product, there's only 3% of target cells. And after the enrichment on the Clinimax Prodigy, we end up with 30% of Tregs. So the next step would be the labeling with max GMP fluorescent antibodies. And please keep in mind that all of these steps are automated. And after that, also the cells will be released into max GMP buffer to go on with the sorting process. And here is an example of this automated labeling. So please keep in mind that we do offer already a predetermined panel for this process, and it is based on max GMP fluorescent antibodies, but it is also possible to get customized panels and to optimize the cell sorting according to your needs. For this, just get into contact with our experts. So now let's have a closer look at the max quantitative cell sorter. This is a multi-parameter flow sorting device. It has 3 lasers and 8 fluorescence channels and the scatter channels. Everything is really happening in a closed cartridge that you see on the top here on this slide. Everything is sterile. The system is an easy plug and play system, and it's also easily scalable. Because we use a closed cartridge here, there's no carryover and also no cleaning needed because there are basically no fluidics in the instrument itself. On top of that, there's also no droplet or aerosol production by the instrument. And samples are never lost in the title. They are all regained in the cartridge. So let's have a closer look at the cartridge. There is the input chamber where the cell product is coming into. Cells are kept in suspension with this mixing propeller, and the cells that are target cells are sorted into the positive collection chamber, and all of the other ones, they would go to the negative collection chamber. So as I said, there is no waste on the Max Quantito. All the cells are kept in this cartridge. The real magic is happening then on the bottom in the microchip. And here is a microchip where the actual sorting process is happening. You can see how small this microchip actually is. And we will also have a look at a short video to look how the sorting process is actually working. So here's a valve that can move. It can move really fast, up to 30,000 times per second. And when the sorting process is active, the green cells here in this example, they are sorted into the positive collection chamber. So the Max Quantito cell sorter is fully closed and sterile. There are no fluidics inside of the instrument itself. Currently, it is being used in more than 10 different clinical trials in phase 1 or also phase 2. And until now, over 200 patient samples have been sorted. All the consumables are available in Max GMP grade. So the GMP Tito cartridge, the GMP Tito running buffer, as well as the Max GMP fluorescent antibodies. This means that everything touching the cells is actually GMP compliant. And we also do provide the documentation for that. So let's have a look at some data here. So let's start from left to right, and we have the product here after the prodigy enrichment. We end up with 38% of Tregs, and then we go into a two-step sorting. First, a debulk sorting is done, which is achieving 83% of pure Tregs, and then the purity sort, we will end up with 98% pure Tregs. So let's move on to the next step, which is Treg expansion. So if you have any special requirements for the Treg expansion or want to have your workflow automated, there are also customized solutions. Please get into contact with our experts and the CAP team to get such solutions running for you. So Treg expansion is a really crucial step and also very challenging. So here we do have, after the sort, the Tregs, we are staining for FOXP3 to check if this marker is still expressed. And then there is a 14-day culture done in Tex-Mex medium with stimulants. And after the expansion, the cells are again stained for FOXP3 and still stably express this Treg marker. But please don't forget that Treg expansion is really donor dependent, and it's also depending on the manual protocol that is being used. And of course, also the expansion duration plays an important role. So the last step is the Treg analysis. So Treg analysis can be done in two ways. On one hand, we can do a phenotypical analysis, and on the other hand, we can also do a functional analysis. Here are different markers named. First, one can do the basic Treg identification, but there are also additional markers possible that further characterize the different Treg subsets. For the phenotypical analysis to support you, we do offer different options. We have the Treg detection kits. These are optimized antibody kits that are pre-titrated already, and it's also a one reagent solution to really speed up the process and make it as easy as possible. The second option are our affinity recombinant antibodies and biodi antibody conjugates. These are high purity and lot-to-lot consistent reagents. And with these recombinant antibodies, it's also not needed to do any Fc receptor blocking steps. And to make it even easier, there's only one universal isotype control. The functional analysis of Tregs is a cellular assay, and for this we also provide a kit, which is the Treg expression inspector, and this is based on MaxiB technology and coupled to antibodies, which gives a polyclonal stimulus. And how does it work? So in a co-culture with responder T cells and MaxiBeads, the responder T cells would happily proliferate according to the stimulus. But when we add the Tregs on top, because Tregs are suppressing immune responses, they would then also suppress the proliferation of the responder T cells if they are fully functional. So these are the main three steps here: the Treg isolation, we have talked about the Treg expansion, and we also defined different options for the Treg analysis. And with this, I'm almost at the end of the talk, but at the end here, I also want to remind you that we have, of course, much more information. We offer brochures for the different products. We do have scientific reports and also posters. We have application notes that are explaining the different steps of these applications and showing data, of course. There are also comprehensive reference lists if you are interested in further reading. And I can really recommend to have a look at all this information here. And with this, I really want to thank you for your attention. And I'm really happy that you were here listening to this presentation.
Thank you again, Doctors Hengst, Salinas, and Steinbach, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.